Hello everyone. The topic given to us is the nervous system and the subtopic is sensory physiology and coding in the nervous system. The team members are Joy, Samreen and Raotong. Let's go back to the basics that we learned in class. What is a synapse? The site of transmission of electrical nerve impulses between two neurons. In a synapse, thousands of vesicles with neurotransmitter are transmitted to the neighboring neuron, and this causes the neuron to depolarize or hyperpolarize. And these two phenomena occur when signal is transmitted from a sensory receptor to the central nervous system or vice versa. The synapse can be of two types. One, an excitatory synapse, which makes the receiving neuron to elicit an action potential. Second, an inhibitory synapse, which reduces the chances of action potential in the neighboring neuron by initiating an inhibitory postsynaptic potential. The paper which we have chosen is Restoring Neuronal Chloride Homeostasis with Anti-NKCC1 Gene Therapy Rescues Cognitive Deficit in a Mouse Model of Down Syndrome. And the reason behind choosing this paper is we were able to learn a lot of concepts in depth, such as inhibitory synapse, that is very important for sensory physiology. This paper also helps us to learn in detail about the pathophysiology that could occur in the central nervous system if chlorine homeostasis is disturbed. The first author of the paper is Martina Parini. She is currently a postdoctorate researcher in Istituto Italiano di Tecnologia. She studies synaptic plasticity and retinal degeneration. She has 8 research works with 266 citations and 2,439 reads. The corresponding author of this paper is Professor Laura Cancida. She is a senior researcher tenured at Istituto Italiano di Tecnologia as well. And she is a principal investigator of the Brain Development and Disease Laboratory. The Electrophysiology Laboratory has 2 setups, one for ex vivo electrophysiological recordings and one laboratory for behavioral tests of young rodents in their home cages. Let me get into the introduction section of the paper. Optimal neural network activity and brain functions critically depend on the correct balance between excitatory and inhibitory synaptic inputs. Gamma-aminobutyric acid is an amino acid that functions as the primary inhibitory neurotransmitter for the central nervous system, and chlorine ion is responsible for the inhibitory synapse. There are 2 types of GABA receptors. GABA type A receptor is an ionotropic chloride channel that mediates fast inhibitory signals, and metabotropic GABA B receptor produces slow and prolonged inhibitory signals via G protein and second messengers. In mature neurons, low in intracellular chloride concentration causes inward flow of chlorine ions through GABA A receptor, causing the neuron to hyperpolarize. However, relatively small changes in the neuronal chloride ion can sufficiently switch the polarity of the GABA A response from hyperpolarizing to depolarizing, as seen during development of rodent from neonatal to postnatal stage. Regulation of neuronal chlorine ion is mainly controlled by the opposing action of 2 major cotransporters, the chlorine importer NKCC1 and the exporter KCC2. In NKCC1 pump, 1 sodium, 1 potassium and 2 chlorine ions are imported, and in KCC2 pump, 1 potassium and 1 chlorine ions are exported.
So what is the importance of chloride homeostasis and regulation of importers and exporters? How are they relevant to physiology, medicine, or to the field of bioengineering? In immature neurons, high chloride ion concentration causes depolarizing effect and plays an active role in neuronal growth and the formation of synaptic connections. As the neurons mature, a development switch occurs, causing a hyperpolarizing or shunting response generated by the GABA signaling in the presence of low chloride ion concentration. If the chloride homeostasis is altered due to variations in the expression or transport activity of NKCC1 or KCC2 transporter, it can give rise to different neural disorders such as schizophrenia, Down syndrome, autism spectrum disorder, epilepsy, Huntington's disease, and many others. Again, chloride ion homeostasis is important in establishing the GABA polarity switch for proper neuronal network formation in bioengineered neuronal organoids, which are promising tools for disease modeling and drug development. One of the current prominent approaches for addressing the chloride-ion imbalance in neurons is a pharmacological treatment that involves the administration of FDA-approved NKCC1 inhibitor bumetanide. Bumetanide has been found effective in rescuing brain-related core deficits in rodent models having a number of neural disorders, as well as in clinical trials with autistic individuals and some case studies on patients with other neurological disorders. However, this treatment has a few drawbacks. One of them is that it requires the daily intake of the drug. Again, bumetanide has a strong diuretic activity that can trigger ionic imbalances and potential ototoxicity, which is unfavorable for lifelong treatments and can jeopardize treatment compliance in the long run. Also, a strong and demonstrated direct link of bumetanide to NKCC1 inhibition and restoration of chloride-ion homeostasis is yet to be known. Taking into account the limitations of the current treatment approach, this research focuses on a permanent solution of establishing chloride-ion homeostasis in neurons at once by NKCC1 knockdown through RNA interference. RNA interference is a rapid method of silencing gene expression where small interference RNAs or microRNAs are introduced into the cell exogenously through vectors or endogenously respectively. This forms RNA-induced silencing complexes which afterwards pair up with a complementary sequence in an mRNA molecule and degrades it, inhibiting the synthesis of a specific protein. In this research work, 2 artificial microRNAs were designed using 2 different target sequences against mouse NKCC1. These artificial microRNAs were first cloned into a plasmid and then subcloned into lentivirus vectors and adeno-associated virus vectors. The vectors were then transduced into the neurons of TS65TN mouse having Down syndrome.
So the first result is that anti-NKCC1 AMI treatment restores intracellular chloride concentration in diseased neurons in culture and in vivo. So the first 2 graphs are experiment done in culture. We can see that in the control group, the diseased neuron have a much higher NKCC1 concentration compared to the wild type. However, after the treatment, both wild type and the diseased neuron all show significant decrease in concentration of NKCC1. When we are looking at chloride concentration, the high value of MQAE fluorescence indicates a low concentration of chloride. So we can see that after the treatment, both wild type and the diseased neuron show decrease in chloride ion, and it's the same with the last graph, which is in vivo experiment. The wild type and diseased neuron all decreased in NKCC1 concentration after the knockdown treatment. The second result they show is that anti-NKCC1 treatment restores hypopolarizing GABA-A signaling in diseased neurons in both in culture and in vivo. So in this experiment, they used bicuculline, which is a type of GABA-A antagonist. So when use this type of molecule, the inhibitory GABA-A neurotransmitter won't be able to work. So this means that there will be more spiking. So we can see that in wild type during both in culture and in vivo, they show high level of spiking after using the bicuculline. However, in diseased neurons, it actually shows a decrease in spiking, which is abnormal. However, after the anti-NKCC1 treatment, the diseased neuron also show a increased level of spiking for both in culture and in vivo. The third result is that neuron-specific anti-NKCC1 treatment in vivo rescues learning and memory in diseased mice. They did 4 tests to assess the learning and memory ability in diseased mice. We can see that before treatment, all diseased mice have bad performance on all 4 tests, which are represented by red. After treatment, which are represented by yellow dots, they all improved their score significantly. What we would do differently is that we want to explore the anti-NKCC1 treatment on developmental neurons. So during the whole paper, they mostly work on mature neurons. However, Down syndrome is the most common genetic chromosomal disorder and cause of learning disabilities in children. And the paper also mentioned that high level of chloride ions also occurs during developmental stage in wild-type mice. So we think it would be interesting that we implement the gene therapy treatment on developing diseased mice and compare the results with the developmental wild-type mice. And I think it would generate results that has high implication on whether this sort of treatment can also help treat children.
Thank you.